First of all, just want to thank the organizers, Alex and company biologists, for giving me the opportunity to speak today. Yeah, and so I will continue a regeneration and ECM theme by talking about an animal capable of whole body regeneration, Hydra vulgaris, and the role that ECM remodeling plays in that process. So, if you are not familiar with Hydra as a model organism or the work in the Giuliano lab, we are focused on this small freshwater cnidarian that has a really simple body plan consisting of a head, mouth, and tentacles on one end and a foot on the other. And those are separated by this tube-like body column. And most importantly, for my talk, the Hydra is capable of whole body regeneration from fragments of tissue as small as a couple of hundred cells. So if you see this video here where time is measured in hours and minutes, within about 2 days, you see the reformation or the regeneration of head, tentacles, a mouth, and the beginning of the regeneration of a foot as well. So an animal that's capable of such astonishing feats of regeneration should be able to tell us some really interesting things and answer some fundamental questions about the cell biology of tissue regeneration. And the one that I am particularly interested in is the role that extracellular matrix plays in that process, namely, to what extent is the ECM remodeled and how is it remodeled? And then what role does that remodeling play in the renewal of lost cell populations? So another reason that Hydra is a nice model for studying this question is that its body plan kind of just has the bare bones for what you would need to study the role that ECM plays in regulating tissue interactions. It has just two epithelial layers. Here you see the ectoderm and endoderm represented in green and magenta, respectively, and they are separated by a continuous ECM. Here I'm marking collagen 1, but the Hydra ECM also has proteins like collagen 4 and laminin. And if we look at a cryosection through around this region of the animal, you can again see that you just have these two epithelial layers separated by the ECM throughout the entire animal's body. Further, cnidarians occupy a really interesting place in the animal kingdom with respect to studying the evolution, evolution of the ECM as well as many other molecules and genes and processes. So, a lot of the core components of the ECM arose at the dawn of Metazoa. They were really crucial to the dawn of multicellularity, of course. So these are things like fibrillar collagen, laminin, matrix metalloproteases, and other ECM proteases. As cnidarians split off from bilaterians, bilaterians continued to add components to the ECM, including expansion of existing gene families, addition of genes like fibronectin, tenacin, and hyaluronin synthase. And cnidarians evolved in parallel and maintained these core components of the ECM. So we can study some of the genes that are most central to metazoa and to multicellularity. So I started out by looking at by using the aforementioned collagen antibody to look at what was happening to the ECM during head regeneration. So bisecting the animal in the middle of the body column and looking at head regeneration. And what I found was that there's actually this ECM depleted region, a gap after the wound closes in collagen localization. And we can be sure that this is in fact a gap in the ECM. It's not an acellular region or a failure of the wound to close. This wound is really closed and it's just lacking collagen. And this is also true of laminin. I'm showing just the collagen antibody for simplicity's sake during this talk. And not only is that gap formed early on, it's maintained for quite a long time and even expands as head regeneration proceeds for the first couple of days, which we can see quantified here. So this gap in the of ECM protein localization in the regenerating head expands over the first couple of days post-injury. Because whole mounts are not necessarily great for looking at protein quantification, I turned to cryosections of these animals to get a better feel for exactly how much these collagen and other proteins have been depleted. So if we look at just the amount of collagen fluorescence in the regenerating head over time, here shown in roughly in this boxed region, and compared it to the uninjured animal, we can see that fluorescence in the head compared to the rest of the body column down here is significantly reduced through the first couple of days post-injury. In some cases, less than 10% of what you would see in the uninjured animal. And it takes up to 48 hours even to get back to half of the complement of what you would see in an uninjured animal. So this is a really extended period in which the extracellular matrix is depleted in the regenerating head. Given that, I've shown here that when you injure the animal, bisect it, you get a very quick wound closure, but the presence of a gap in the ECM that actually grows significantly over the first couple of days post-injury. And so I was interested in how this is broken down and then how it's built back up again as regeneration is completed. To answer that, I first turned to matrix metalloproteinases. They're one of the best studied, most conserved families of ECM regulators and ECM regulating enzymes, proteins involved in the breakdown of ECM. So I found this drug merimostat, broad spectrum MMP inhibitor, and found that with continuous treatment in the early stages of regeneration, you see this inhibition of the animal's ability to make that collagen gap, which we can see quantified here, a significantly shorter gap in the animal. And not only that, but if we continue drug treatment to the later stages of regeneration, when we actually start seeing a new head and tentacles form in a control animal, this drug treatment does significantly delay head morphogenesis as well, and see that there are much fewer tentacles formed when the animal is continuously treated. So from there, I wanted to look at, I mentioned this is a broad spectrum inhibitor, so I wanted to look at actual candidates. And thankfully, we have a pretty good transcriptome of the Hydra due to earlier work by our lab mate, some of my colleagues. And I found initially a dozen genes with uniprot blast hits to various MMP family members. And with some help from a postdoc in the lab, found that 11 of those 12 have at least a couple of the domains that are conserved in MMPs. And so I looked at their spatiotemporal expression and looked at tissue localization through fluorescence in situ hybridization, and found that 5 of these MMP-like genes are actually expressed during head regeneration. So here's one of these genes that's expressed throughout the body column in uninjured animals, but then is significantly upregulated in the regenerating head. And we see about 4 other genes that have exactly the same tissue expression pattern, where the head regeneration sees a significant increase in expression. Not only that, but all of these genes are expressed primarily by the endoderm. So it seems that the breakdown of ECM is primarily an endoderm-driven process. So from looking at the breakdown of ECM, I wanted to see how it's built back up again to complete regeneration. And to address that, I tried another drug called dipyridil, which is a collagen cross-linking inhibitor. Stephanie introduced the collagen at the beginning of her talk, but collagen is created by these triple helices that undergo cross-linking to make the mature protein. This drug inhibits that, and we can see that there's a significant decrease in the head fluorescence when you treat with this drug. So we know it is indeed inhibiting the formation of new collagen fibrils. And when we treat for longer periods, we see a complete blockage of head regeneration, and also this animal tends to kind of balloon up because you're not making new collagen. The tissue is fragile and very easy to stretch. So again, total blockage of head regeneration when we block collagen cross-linking. We also wanted to look at where the new collagen is coming from, because there are a few older papers on this subject that implied or stated that new collagen is being made by the ectoderm. That disagreed with some of the single-celled sequencing data that we've published, and we indeed found that in the uninjured animal, endoderm is making collagen, and in the regenerating animal as well. So that could have just been a bit of an issue in interpreting older colorimetric in situs, where it's a little bit harder to accurately define localization. But the takeaway here being that the same tissue is responsible for expressing the genes that break down collagen and also make new collagen. So when you have this injury, you get a quick wound closure and formation of this ECM gap, and MMPs are expressed as the gap grows, but also we see the expression of collagen transcripts, and because these are expressed simultaneously, there has to be some kind of very tight coordination, choreography at the post-transcriptional and post-translational levels to make sure that this breakdown and buildup of collagen occurs at the correct times. And then lastly, what I was really interested in how is this affecting stem cell behavior? So in addition to these two layers I mentioned, Hydra actually has a third cell population, the interstitial cells and their derivatives. So interstitial stem cells are located exclusively in the ectoderm, but their derivatives, which include gland cells, neurons, germ cells, are located in both cell layers. And it's been described before in homeostasis through some cell labeling that these cells have to invade, but it's never been observed and it's never really been studied during regeneration. However, we know that when the animal is bisected, it loses a bunch of head-specific endodermal cell populations, namely these two types of head gland cells that are responsible for secreting mucus. So you lose these entirely when the animal's head is cut off, and they need to be replenished somehow. So my hypothesis was that perhaps this big ECM gap, this extensive ECM remodeling, plays a role in facilitating the invasion of these interstitial stem cells to replace lost cell populations. So here we see these interstitial stem cells marked by nanos promoter driving GFP expression. They're expressed all along the length of the body column, but excluded from the head, tentacles, and foot. When we look at cryosections of the animal, again, we can see these are in the very, very densely distributed in the ectoderm and not at all in the endoderm, and they're largely excluded from the head as well in an uninjured animal. When we look at early in regeneration, we can see that these interstitial stem cells are in the regenerating head ectoderm, but still largely excluded from the endoderm. But as we go through the time course of regeneration, we see more and more of them accumulating in the head endoderm, which we can see quantified here in just a second. But what I wanted to draw your attention to is there's a very clear ECM depleted region here when you compare collagen intensity to what we see here towards the bottom in the uninjured body column. And we're seeing those nanos GFP positive cells accumulate in the endoderm at these sites of low ECM expression. And indeed, between the first day and day and a half, you see a pretty large jump in the expression, in the localization of nanos GFP positive cells in the endoderm. And that's happening around the same time that there is still a very low complement of ECM in the regenerating head. So the working model I have thus far for the role of ECM in Hydra head regeneration is that decapitating the animal, bisecting it at the midpoint of the body column, obviously causes this huge tissue injury, but also removes head-specific interstitial stem cell interstitial derivatives. You then close the wound, see the growth of this ECM gap, and at the same time, invasion of these interstitial stem cells to replace lost interstitial derivative populations. So with that, I just want to thank my entire lab, people, specifically Abby, Stefan, Jack, and Angel, who helped with this project, as well as Ming Zheng for providing the collagen antibody, SDB, and the Hartwell Foundation for funding. So yeah, happy to take any questions. Thanks again for the opportunity to speak.
Wonderful. Thank you very much, Ben. Please do ask questions using the Q&A box. And while people are typing things out, I'll start with a question. So you focus here on collagen, but you also mentioned at the beginning that laminin has a similar expression. Do you know if laminin's also remodeled in the same way during regeneration, and is laminin derived from the endoderm cells as well?
Yes, we can tell from our I mean, I've done the in situs on it, and we can tell from our single cell data that yeah, endoderm is making laminin too. So it really seems like endoderm is in control of like almost this entire remodeling process. And then as far as the dynamics, they're pretty similar. It does seem like there's been a little work done on this before. Uh, some papers from Michael Sarris's lab. And it seems like the laminin undergoes somewhat similar dynamics, but does appear to get filled in a little bit faster. So I think it's possibly being made somewhat faster than
Thank you.
the collagen, but pretty similar dynamics.
Thank you. So we have a question from John Pugh asking, is there a comparable situation for the regenerating foot?
Yeah, so I'm really interested in looking at that because I just focused on the head thus far because of the interesting localization of the head specific endoderms. You do endodermal cells. You do see a gap in the foot. I think it's probably not quite as big. I have not actually like tried to rigorously quantify it yet. But I'm hoping to look into that soon, especially because a former postdoc in the lab who now has his own lab back in Mexico, he has been really interested in studying in comparing regeneration in vulgaris and Hydra, which is a closely related species that underwent an evolutionary loss of regeneration in the foot. So it can still regenerate its head normally, but most of the time fails to regenerate a foot. And he did some transcriptomic work on that, and it does seem like there are some changes to ECM in some basically he showed that you can rescue foot regeneration by overexpressing Wnt in these animals that fail to regenerate normally, and when you overexpress Wnt, you get this large upregulation of ECM and ECM remodeling proteins. So it's possible that there's some evolutionary role for ECM remodeling in rescuing that normally failed regeneration in the foot. So that's something I'm really interested in following up on.
Okay, thank you very much. Another question from John asking whether the collagen layer is responsible for preventing the migration of the interstitial cells.
Well, it is. So I think it's more of a like balance kind of. There could be too much or too little. Obviously, you need collagen as a substrate, and there have been some papers in the past that show that the ability of these cells to migrate, these interstitial cells to migrate at all, is dependent on collagen. So they need it as a substrate. What I think is happening is just that normally the ECM is sort of thick and rich enough that you don't see large amounts of invasion through it. It's kind of similar to what you would see in other models of cell invasion, like the C. elegans anchor cell model. But because there is such a need for cell replacement during head regeneration, it can be facilitated by this breakdown of ECM. And as far as there's sort of two parts of that, like the migration of them maybe to the head and the invasion from one epithelial layer to the other. And the thing about their migration is that when you cut the body column, when you cut the animal in half, you now actually have stem cells kind of already present there. They're not, those stem cells are not normally present in the head, but when you cut the animal in half, the midpoint of the body column will become the new head, and there are already the stem cells there. So there's not as much of a requirement for active migration up to the head. But so that's not really affected by the depletion of the ECM. But I do think it facilitates invading quickly from one epithelial layer to the next.
Thank you. We have a related question from Stephanie, who's asking, so the ECM layer seems to be a physical barrier between the endoderm and the ectoderm. And in limb regeneration, that's broken down to facilitate signaling between the two germ layers. Is a similar situation happening here? Do you see an increase in growth factors being expressed in one tissue that are signaling to receptors in the other tissue?
Yeah. The signaling, sort of upstream and downstream signaling components, are definitely something I'm like really interested in studying more going forward. I haven't really done it in this particular study, although it is known that um, other people have done some work on the earliest sort of signaling responses to injury. Namely, for one, we know that sort of Wnt is kind of the master regulator. It almost has like sort of the opposite effect that it does in planarian regeneration. So it controls head regeneration as opposed to the tail regeneration in planaria. And it is expressed in both tissue layers, Wnt that is, during regeneration. So it's possible that that one of the things that the ECM depletion is doing here is making it so that it's very easy to transmit those signals to both tissue layers. So that's certainly a possibility, and there's been other work done, including some work going on in my lab right now by a grad student. Well, not my lab, Selena's lab, by a grad student, Iris. She's really interested in looking at ERK signaling, which is activated very quickly, within minutes of injury, and it does light upstream of that transcriptional increase in Wnt. So it's definitely possible that some of this depletion in the ECM is facilitating crosstalk between the two epithelial layers.
Did you want to follow up on that, Stephanie? No.
Thanks.
Well, thank you.
We have two final questions. Hidehiko Hashimoto is asking, well, disassembling and reassembling an ECM is very expensive from a metabolism point of view. They're very large macromolecules. So he's saying that the ECM degradation is important, but why is the breakdown necessary? Could the ECM structure is the original ECM structure unusable for the assembly? I guess, are the ECM components being recycled in any way?
Yeah, so I'm not positive how much of this is recycling versus completely new synthesis. Obviously, we know that there is a large transcriptional upregulation, which we can see that just through visualization methods like FISH, but also through transcriptomics, both some of the early our lab is mostly focused on some of the earliest stages of regeneration, but there are other labs like the Galileo lab that have done like the later portions, and you do see a sustained transcriptional upregulation in these ECM components. As far as whether some of them are whether some of that ECM is recycled, I haven't actually done those experiments to directly look at maybe what the proportion of new synthesis versus recycling of existing ECM is. But yeah, that's certainly a very good question, because these are huge macromolecules that are pretty heavy duty to be breaking down and building back up.
And to follow up on that, do you have any idea whether the composition or the organization of that first extracellular matrix is different after it's been regenerated or reassembled?
Yeah, so that's one of the things I'm really interested in doing, like writing in research statements right now as I apply for jobs. Because so there's this idea throughout. I mean, I kind of asked this question to Stephanie already, but like this idea that there's sort of something that's often called in the literature like a transitional matrix, which is that during regeneration the composition of the ECM changes. And you might see downregulation of collagen 1 and upregulation of like other collagen subtypes, that sort of thing. And so far, all we know is because we have these antibodies, there's this depletion in both collagen 1 and laminin. But whether we see other changes to the ECM, we don't know yet. And some of the there have been a couple proteomic studies on the Hydra, but they either look too early, like they're only looking at the very, very earliest stages post-injury, or they're like just sampling the entire animal. And so what I want to do is look a little bit later and also enrich for the ECM using some decellularization methods. And so that's one thing I'm really excited about looking for.
Cool, thank you. And a final question from Emerald Perlas. I'm going to read this out verbatim because I'm not sure I totally understand. Could the new collagen layer be set by the invading stem cells instead rather than by the interstitial cells at the injured site?
Sorry, could you repeat that real quick?
Could the new collagen layer be set by the invading stem cells instead rather than by the interstitial cells at the injured site? Do you see any
Yeah,
expression of
so
collagen by these stem cells?
it we don't usually. So like, I mean, we have the antibody, we have the ability to look for transcriptional upregulation, obviously with fluorescence in situs. We don't tend to see much collagen expression in the invading stem cells.
Mhm.
Some the interstitial stem cells, some of their derivatives do make these what are called mini collagens. They're expressed in nematocytes, and so they're just like these much smaller versions of collagens that we can pick up with the antibody as well. But those are more in like slightly differentiated nematocytes. Those are the stinging cells that make cnidarians. They're yeah, the cnidocytes. So those will express some collagens, but we don't tend to see much by the invading cells themselves. And that's again sort of an interesting like basis for comparison in some of these with regards to like how this remodeling process works. Because like with MMPs, for example, like you can see in other regeneration models that sometimes the breakdown of ECM is driven by invading cells themselves, and other times it's driven by sort of support tissue that's nearby. And pretty much um every case of like a metalloprotease expression that we've seen, it appears to be the latter in the Hydra, that that nearby tissue, namely the endodermal epithelium, is expressing all of the genes involved in both the breakdown and buildup of new ECM, whereas the interstitial cells don't really seem to be expressing much of the many of the genes that are responsible for that.
Sure. Thank you very much for all your answers. Fantastic talk. Thank you to both our speakers. A virtual round of applause for you both. This is our last Development Presents webinar 2025, but we'll be back in January next year. Thank you very much, everyone, and bye for now.